Our article explores the phenomenon of RNA interference and some of its key components, specifically a member of the Argonaute protein family dubbed as AGO2 or AGO2. To preface, epigenetics refers to heritable changes not encoded by DNA. These changes affect gene expression by turning genes on and off. Our article targets RNA-mediated epigenetic regulation of gene expression via RNA interference, also known as RNAi. First, we will explain what RNAi is from an up-to-date description. RNAi is a process where sequence-specific gene silencing is induced by double-stranded RNA triggers. There are two types of RNAi, microRNA, also known as miRNA, and short interfering RNA, also known as siRNA. miRNAs are responsible for multiple messenger RNA recognition, but for this experiment, highly specific siRNA are the main components used to suppress mRNA. siRNAs are double-stranded RNAs cleaved into 21 to 25 nucleotides by an enzyme called dicer. Then an Argonaute protein binds to the siRNA and discards the sense strand. From there, Argonaute in the antisense RNA strand forms RISC, or an RNA-induced silencing nuclease complex. The RISC complex base pairs complementarily to a target mRNA strand to inhibit its translation or to degrade it. Both results in gene silencing. Traveling back to 2001, this article describes the purification of RISC from cultured Drosophila cells and reports the identification of an Argonaute protein within the enzyme. This subunit, named Argonaute2, begins linking the genetic analysis of RNAi from diverse organisms and the biochemical model of RNAi from Drosophilus in vitro systems.
The researchers' objective was to identify the proteins and RNAs that carry out RNAi in vitro as a step toward unifying biochemical and genetic data into a single mechanistic model that predicts real-world phenomena. As mentioned, RNAi is mediated by the RISC complex and is known to contain siRNAs. However, the protein components of this activity are unknown. Thus, experimenters sought to report the biochemical purification of the RNAi effector nuclease from cultured S2 Drosophila cells or fruit fly cells. Researchers used S2 cells as they are phagocytic and readily take up DNA from the cell culture. This allows for high co-transfection rate and subsequently a high expression rate of multiple proteins. Future studies reveal how Argonaute is able to selectively identify target RNAs. Figure B shows the seed region in which guide RNA is cradled into a hydrophilic channel centered between two protein lobes that are held in a helical conformation. Argonaute then makes direct hydrogen bond contacts only to the phosphate backbone, leaving the base edges solvent exposed.
Scientists wanted to first purify RISC by separating it from fruit fly cell extracts. These cell extracts were transfected with a luciferase double-stranded RNA so that they could analyze which part of the nuclease complex contains an active role in RNA interference. Previous studies indicate that RISC is attached to ribosomes and cell extracts, so the first step of the purification method is to centrifuge out the ribosome pellet and wash out the supernatant. Next, a sequence of high and low salt extractions further purify the ribosomal proteins. After the protein was purified, the superrow 6 column, which is the size exclusion chromatography, was first to run. This separated the ribosomal proteins into fractions and was assayed to determine luciferase mRNA and a control mRNA expression.
Analyzing the figure, we can see RISC silencing activity above 440 kilodaltons, which shows the least amount of luciferase expression, and it corresponds with the actual measured size of RISC, which is 500 kilodaltons. The fraction containing peak RISC activity in the superrow 6 column, the one with the least expression of luciferase, was then loaded on a mono S column and assayed. This was done in the same manner for the mono S column being placed into the mono Q column, both of these being ion exchange chromatography columns. This was repeated yet again by taking the fraction with peak RISC activity of the mono Q column to the hydroxyapatite column and was assayed. The fraction of peak activity of this column was then run through an SDS page gel, which separates proteins by size, and the results yielded the complex wasn't fully purified, as it showed 4 bands. The different sizes indicated that there were proteins interacting with the RISC fraction.
Scientists excised candidate proteins from the SDS gel and microsequenced the proteins using tandem mass spectroscopy. This resulted in 2 viable bands of 87 and 130 kilodaltons. Upon further analysis using cDNA libraries and PCR, researchers confirmed a 130 kilodalton peptide. Database and domain database searches identified the protein as a homolog of RDE1, which is a member of the Argonaute gene family that proves essential for RNAi in roundworms. Thus, researchers named the gene Argonaut2. The Argonaut family is comprised of at least 4 members, including Argonaut2, but so far only AGO2 has been detected as a component of risk, though it is possible other Argonaut family members join the risk complex at points. After sequencing AGO2, scientists believed that the 5' end of the predicted sequence was missing. This motivated researchers to construct a gamma-zap library from RNAs from Drosophila embryos and S2 cells to identify AGO2 cDNA clones.
Sequencing these clones revealed discrepancies with predicted AGO2 gene, with predicted introns appearing in the mature mRNA, which was confirmed by northern blotting. This 5' extension was primarily composed of 11 63 nucleotide direct repeats, which translate into a glutamine-rich region. Similar regions can be found in Arabidopsis AGO1, but not in Drosophila AGO1. AGO2 also contains PAS and PIWI domains. However, similarly to the polyglutamine domain, the functions of each of these are unknown. The fractions were assayed for risk activity, and scientists were able to verify the presence of AGO2 in risk by generating AGO2 specific antibodies, followed by Western blot analysis. Specifically, scientists created AGO2 tagged with T7 epitope and polyhistidine chains at the N terminus to express the gene with induced RNAi for luciferase. Figure C shows that fractions 17 and 18 have degraded luciferase transcripts with corresponding bands for the presence of AGO2. In addition, risk was affinity purified on a polyhistidine binding resin. After analyzing the imidazole elution by Western blotting with a T7 antiserum and northern blotting with a luciferase probe, interaction between AGO2 and 22 nucleotide siRNAs were found, pointing to interaction between luciferase and endogenous AGO2 and risk. This data further supported that AGO2 is a component of risk.
Once the presence of AGO2 in risk is verified, the next logical step is to check if AGO2 actually affects RNAi. This is where the experiment gets a little meta, where the researchers suppress endogenous AGO2 through RNAi, suppressing the suppressor, if you will. The experiment involved treating Drosophila S2 cells with dsRNA templates of the first 500 nucleotides of GFP and 2 1000 nucleotide homologs of AGO2 at 5 and 3' ends. The Western blots of these reveal suppression of AGO2 protein in lanes 2 and 3, while the control GFP was expressed as evident in the band in lane 1. The extent of suppression of AGO2 was further studied by transfection with a mixture of firefly and vanilla luciferase expression vectors, with dsRNA triggers of GFP as a control and firefly luciferase. The ratio of firefly to vanilla activity in graphs 2 and 3 suggests the suppression of AGO2. Silencing of AGO2 would mean more luciferase in the cells, and the ratio of firefly to vanilla would be higher, considering that the exogenous reporter vanilla luciferase is the same amount. The 5' dsRNA trigger is suppressing AGO2 more than the 3' trigger. As of this study, the biochemical functions of the domains of the Argonaut family were unknown. However, AGO2 did share the PAS domain with dicer, and because dicer processes dsRNA to siRNA, the researchers considered that these complexes might physically interact. To test if that is the case, the researchers T7 epitope tagged the dicer protein and immunoprecipitated the transfected S2 cell lysate with a T7 antiserum. In the eluted mixture is possibly dicer with AGO2, which was then treated with affinity purified anti-AGO2 and analyzed by Western blot. The figure on the left with a band on the blot verifies the interactions between dicer and AGO2, proving that they interact with one another. This also leads to 2 thought processes regarding dicers. 1, dicer is a component of risk but fails to process dsRNA triggers when associated within the complex, are the current model, which suggests that the interaction between dicer and AGO2 processes siRNA into risk, which then dissociate from dicer and target specific mRNAs to degrade.
During the time of this research, the function of the PAS domain was still unknown. However, research done in 2012 suggests that the PAS domain binds the 3' end of small RNAs. Further questions include, are the roles of Argonaut, which include gene silencing phenomena and the development of diverse species, mechanically related? And are other members of the Argonaut gene family involved with RNAi? In conclusion, in the process of purifying and studying the RISC complex, scientists were able to identify a member of the Argonaut protein family. Upon further analysis of this protein, they concluded AGO2 is indeed a subunit of RISC that interacts with siRNAs to facilitate RNAi. They had also considered the possibility that the enzyme dicer and AGO2 might physically interact through their shared PAS domain. Thank you so much for listening.